So now let's talk more in detail about what gives skeletal muscle cells that striated appearance, the structure of sarcomeres, and how that relates to muscle contraction. So again, these are the myofibrils. These are bundles of actin and myosin that are found in a skeletal muscle cell. So the striation pattern is made by the arrangement of the myofilaments. So here's the myofibrils, and then the myofilaments are actin and myosin, okay? So myofibrils consist of sarcomeres that are connected end to end. So this unit right here, remember sarcomeres are the functional unit of these myofibrils. This is a sarcomere, this is a sarcomere. So let me define a sarcomere a little bit better and the components of a sarcomere. Okay, so remember thick myofilaments are myosin, and thin myofilaments are actin. Okay, and then so sarcomeres contain these structures right here, and I'm going to show you these on the next slide because it's a little easier to see. All right, so here, this is called a Z line right here, and it has a Z-like appearance. It's a zigzag appearance. This is a Z line. This is a Z line. A sarcomere is defined as being from Z line to Z line. So this is one sarcomere, and these are connected end to end down the entire length of the myofibrils. So let's talk about what is an individual sarcomere from Z line to Z line. So if you look down here, the pinkish color myofilaments, you can tell that they are thicker than the more purpley blue ones, right? So these thick filaments, these are myosin, okay? The thin filaments, which are more purple or blue, these are actin. So actin, these are myosin. Now this is a relaxed sarcomere, and it's relaxed, I know it's relaxed because of the structures that I see. Now here, right here in the smack dab in the middle, okay, you'll see that there's a line right down the middle. This is called an M line. The M line is made up of proteins that anchor myosin in place, okay? So this is the M line. And think M line is midline, okay? Proteins that anchor myosin in place. The Z lines are made up of proteins that anchor the thin and the thick filaments, the actin and myosin in place, okay? But this M line really helps anchor the myosin in the center of the sarcomere, okay? So this area right here in the middle where you see only thick filaments, only myosin, this is called the H zone. So H zone, okay? So here's the H zone. Now over here where you see just thin filaments, so from the Z line over to here where the thick filaments start, this area of the sarcomere is only thin filaments. This is called the I band, okay? So this is called the I band. Now here, from here to here, this is called the A band. And the A band consists of overlapping, so if you see right here, overlapping thick and thin filaments, okay? So let me look at another. Okay, so here, this is a relaxed sarcomere up here. This is a contracted sarcomere. Let's orient ourselves. Here's the M line. These are proteins that anchor the thick myosin filaments in the center. Here's our Z line right here. Here's another Z line right here, made up of proteins that anchor the thick and thin filaments in place. Okay. Now, if you look from here to here, this is our A band. And the A band consists of overlapping thick and thin filaments. Here's our I band right here, which consists of only thin filaments. Right here in the very center, where you only see the thick filaments, where you only see myosin, this is the H zone, okay? H zone. So H zone, only thick myosin filaments. A band, overlapping thick and thin, so thick myosin and thin actin. I bands, only thin actin, okay? All right, so if you can see an H zone and an I band, then it's relaxed, okay? Down here, so what happens is when a skeletal muscle cell contracts, those individual sarcomere, these functional units contract. And it's not that the filaments, actin and myosin, it's not that they get shorter. They don't. They stay the same length. But these thin filaments, these thin actin filaments from this side slide in towards the M line. And the thin filaments over here, these thin actin filaments over here from this Z line, slide in closer towards the M line. So they're sliding in, they slide past one another, and these thin filaments slide in towards the M line, okay? So if you look right here, you don't see an H zone, you don't see I bands, or you see very, very little I band. The bulk of this sarcomere is now an A band, because remember the A band is overlapping thick and thin filaments, right? Overlapping actin and myosin. This whole sarcomere is overlapping actin and myosin, okay? So if it looks like it's all A band, then it's contracted. Again, these filaments aren't getting shorter, they're just sliding past one another. Here, you see an H zone, and you see prominent I bands. This is a relaxed sarcomere, okay? Okay, so that is the anatomy of a sarcomere. So let's look up close on myosin. So here's our thick, this is myosin. And up here, the little balls that look like Mardi Gras beads, those are actin. Okay. All right, so right here, so thick myosin filaments are made up of these myosin proteins. It's a myosin protein. And each one of these proteins are very filamentous, so rod-shaped. And they have these globular heads that come off above and below, okay? So these are called myosin heads, okay? These globular heads that come up and they extend above and below the thick myosin filaments, okay? So that's myosin, made up of myosin protein. Now thin filaments. So the very light blue, like the lightest of all the colors here, okay? So the lightest. Actin, which are the thin filaments. So thin filaments are composed of actin protein, and they are associated with two other proteins, and I'll talk about those in just a second. But if you look, actin is actually made up of globular proteins, so globular meaning somewhat more spherical. So if you look, it kind of looks like Mardi Gras beads, two chains of Mardi Gras beads, right, that are in a helix form, a helix shape. Each one of those Mardi Gras beads is an actin protein. So a globular actin protein that's linked together like a Mardi Gras bead, and you have two of those strands that are in a helical shape, okay? That is actin right there, okay? That is actin. Now, if you look, the darker purple or blue, whatever color your eyes see, so this thin kind of this more filamentous protein that's associated with the actin, this is called, so this is tropomyosin right here. Tropomyosin. And tropomyosin is going to block the myosin binding sites on actin. So what you can't see right now, or you might be able to see a little tiny bit of it, is on each one of those actin subunits. You'll see like a tiny, slightly pinkish area, but it's being covered by tropomyosin. That pinkish area corresponds to these pink myosin heads. That's where myosin will bind to actin. But tropomyosin is in the way, so this would be a relaxed sarcomere. Again, we also see this would be an H zone where we only see thick filaments, right? They're not completely overlapping with the thin. So this is a relaxed sarcomere. These myosin heads cannot attach to actin because the myosin binding sites on each one of these actin subunits is being covered by tropomyosin, okay? So tropomyosin is a protein associated with actin, and it covers the myosin binding sites on actin. So these heads of myosin cannot bind because those binding sites are being covered by tropomyosin. So if we want a skeletal muscle to contract, we need these myosin heads to attach to the actin binding site, to the myosin binding sites on actin, okay? So if we want contraction to take place, these myosin heads need to bind to the myosin binding sites on actin. So in order for that to happen, we have to move tropomyosin out of the way. How do we do that? How do we move tropomyosin out of the way? So here, this protein right here, it's circled right here. These guys, okay? This protein is called troponin. And troponin is made up of 3 subunits. So if you look at each one of these troponin molecules, you'll see it's made up of 1, 2, 3 subunits. 1, 2, 3 subunits. 1, 2, 3 subunits, okay? So troponin is also associated with actin, made up of 3 subunits, okay? Let's go through each one of those subunits because it helps you understand how muscle contraction can take place, okay? So on this troponin molecule, you're going to have an actin binding subunit, right? Actin binding, which makes sense because troponin is associated with actin. So one of these subunits, its job is going to be to bind to actin. So you have an actin binding subunit. There is also a tropomyosin binding subunit. So one of the subunits is going to be bound to tropomyosin. The third subunit of troponin is going to have a calcium binding site. Okay? So remember, calcium is super important, and a skeletal muscle cell is not going to be able to contract unless calcium is present. And then myosin heads are not going to be able to bind unless we can expose that myosin binding site on actin. So the way that happens is upon stimulation by a motor neuron, calcium gets dumped into the sarcoplasm from the sarcoplasmic reticulum, okay? So the sarcoplasmic reticulum dumps calcium out, and because there is a calcium binding site on troponin, guess what? Calcium is going to bind to troponin. When calcium binds to troponin, the conformation of troponin changes. The conformation meaning the shape of it, right? So it changes shape. Calcium binds to troponin, it changes its shape, and guess what? Troponin is also, remember, it is bound to tropomyosin. So when troponin changes shape upon calcium binding, it pulls troponin pulls tropomyosin out of the way. Okay? So I'm going to say that again. Calcium, upon stimulation by a motor neuron, the sarcoplasmic reticulum is going to dump calcium into the sarcoplasm. That calcium is going to bind to troponin. When calcium binds to troponin, troponin changes shape. When troponin changes shape, it pulls tropomyosin out of the way and exposes those little pink areas on actin. When those pink areas are exposed on actin, because tropomyosin has been moved out of the way, now these myosin heads can bind to the myosin binding sites on actin. And when that happens, you can have muscle contraction. Okay? So, skeletal muscle contraction requires interaction from several chemical and cellular components. I've already described a couple of those for you, and I posted 2 really helpful videos for you to watch, and I will tell you when you should watch those in this video. So contraction results from a movement within the myofibrils in which the actin and myosin filaments, remember, they slide past one another. They do not shorten. The actin and myosin do not shorten. The sarcomere shortens, but the length of the filaments do not. Okay? All they do is the actin is pulled in. So remember back here, the actin on this side and the actin on this side, these thin filaments are just slid towards the M line, shortening the sarcomere, but the length of actin and myosin do not change. Okay, so the myofibrils, the actin and myosin filaments slide past one another, shortening the sarcomere. So remember, in a contracted sarcomere, it's almost all A-band, overlapping thick and thin filaments. When all of those sarcomeres, so those myofibrils are made up of repeating sarcomeres, which are the functional units. When all of those sarcomeres contract, the muscle fiber, the overall length of the muscle fiber will shorten. And because muscle organs are connected to bone by tendons, it pulls on those attachment sites and you get movement of the skeleton.